Hello guys, we are going to start the tutorial how to work with this project I call Talos Principle. It's basically, I'm going to show you. So this is how you basically open the program if you need the PyCharm currently. It's not yet built to be run by itself. So, just a second. We're going to run the program. Here we go. Okay, so let's talk a bit about what we are seeing in here, okay? So, you have an option to load background bed file, a target bed file, data bed file, and to do analyze, go, or clear. So when you do load a background bed, you basically load a bed file. Let me know in the comment if I need to explain in details what is bed file. So in data, we are going to, for example, it's a bioinformatics tool. So for example, I'm interested in methylation DNA. So I'm going to take all the known methylations that used in Illumina, and I'm going to use the list of all of those CPG sites. Okay, so this is my background. And now for the target. In the target, I'm going to use two of my own research files. So I have a file that called common, and another one that is jumping CPGs. Okay, so in target you can upload two targets. Basically, you have to put two targets because it's part of the analysis to ask if two phenomena are happening together or happening separately. So, or something between, which is it doesn't matter. They are not related in any sense. So I want to see if CPGs that are jumping are also common. So I'm putting those two files, bed files. Okay, I don't know why it's running again, but I'll just put them again. Okay. Now, the data is basically in which way we are measuring it. For example, we can keep measuring it by CPGs, which means we don't have to load data at all. Or we can load G notation, and then we're measuring it by the CPGs that sitting on genes specifically. So I'm going to do this case. Now notice that all the files that I use are not necessarily sorted. The program is sorting the files automatically, so you don't have to worry about it. The thing that you need to worry about it that I yet fixed is in the next version there's going to be support for list of CPG sites names without the whole file format of bed, which means that you can have a very text basic list, and you upload it, and it's automatically going to become a bed file in the background of the program, if that makes sense. Anyway, if we're doing Go, you're going to see what would happen, which is basically you're going to get a view that is similar to a genome browser. It takes a bit of time, I'm sorry. It's not very efficient. Still Python, you know. We also need to go to an area when we actually can see a difference. So I'm changing through the text box in here, the position we are looking, the range in the genome we are looking. Again, if you have questions about this, if you're not a biologist or bioinformatician, let me know in the comments. I can do a lot of tutorials to each of the subjects we are talking about. Okay? So you can see in here, this is the background, and we have all CPG sites located in all kind of places. The common are not cannot be seen in here, I guess, or maybe this is part of the common, I'm not sure. The G notation we can see very clearly, this is all of this. And the jumping site, we see like a very clear straight line, which means we have at least two CPGs in the jumping site in this range. Okay, so let's go to analysis. Again, it's going to take time. It's a Python program, I'm sorry. What it's doing in the background is basically to take the... okay, it's a bit complicated, but maybe I will leave it to another day because we also have the results in here. Also, you can see what the program do if you go and check the script. So I recommend it if you are interested. Okay, so let's see. We have observed overlap of 155 genes. Remember, we put data in genes. So most of the genes are coming from target 1, which is the jumping sites, and a little bit less are from the target 2, which is the common, okay? Now let's see what the results are. So there's basically a test to see if we are out of the distribution of the random case, okay? So this is how the random case looks like. This is our null hypothesis. And we can see that our 157 is in the edge of this, the lower edge, which means that our p-value is telling us that our, if it's significant, it's telling us that we are having low amount of sites compared to what we expected if we were in the random case. So it might tell us that there is kind of dependence or undependence, like depletion. You can see the depletion is very low. We get beneath 0.05, so it's good. It's also and the enrichment is very high, which means it's exactly the opposite. If the enrichment was low in the p-value, it means that we're getting enrichment. But if we're getting low in the depletion, it means we're getting depletion. And the two-sided is like the harsher statistical test, and we also get a significance in here. So it's very good, because if you know from the start that you want to check this depletion, you can use this value. But if you don't know and you check both of them, then you have to use the two-sided.